remember what I learned in biology at that time. So if any of this is, let's see, either too simple or over your head, just let me know, chime in. If you have questions along the way, you want some clarification, chime in. But a little bit before I go into that, I want to just talk a little bit about who I am and sort of what my background is. And so my name is Tracy Townsend, and I'm actually a surgeon. I'm an orthopedic surgeon, which means I operate on the entire musculoskeletal system. So everything pretty much from the neck down, so spine, arms and legs, and that includes bones, muscles, tendons, all the joints, cartilage. Maybe you guys have a grandparent who's had a knee or a hip replacement that would have been done by an orthopedic surgeon, or maybe you have a favorite football player who tore their ACL. An orthopedic surgeon would repair their ACL, which is a ligament in your knee. There's just a whole host of different surgeries that orthopedic surgeons do. So before I became an orthopedic surgeon, I went to college and studied biochemistry. Biochemistry was what my undergraduate degree is in. And then I went to medical school after that. And now I'm in my final year of residency training, which is a 5-year program. And so it's a lot of schooling, but it's a lot of fun learning along the way and fascinating experiences. And there are a lot of different routes to becoming a doctor. You can study really anything you want. It doesn't have to be something science related. But a lot of doctors have backgrounds in either biology or chemistry or genetics and things like that. So if you have questions just about that at all too, I'm more than happy to answer them. And so why don't we jump into the lesson? I'm going to pull up my screen here and let me know if you guys can see it.
Yep.
Good, okay. So, and then let me pull up my pen. All right. So this, okay, so we're going to be talking about the, about how DNA, let me write DNA here, goes to, let me pull this up real quick. Okay. How DNA goes to RNA. And we'll talk about what all these terms mean, and how RNA goes to what we call proteins. Okay. So, and actually before we even get into this, just to give you an overview, if you were to zoom really far down into a cell, okay, so we are all made up of, let's take our own human body. Any living thing is made up of cells. And every multicellular organism, like a human, where we have a bunch of different cells, every cell has a nucleus. And within the nucleus, there is DNA. So hopefully that sounds somewhat familiar. And then DNA stands for deoxyribonucleic acid. And each DNA molecule is made up of a nucleotide here. So this is a nucleotide. Okay. And every nucleotide, this is how we represent different structures of molecules in chemistry. So as you guys kind of advance in your science learning, you'll learn more about how bonds are formed between different atoms. And this is how they're represented. And so every nucleotide is made up of a phosphate group here. Okay, and then that is attached to what we call a sugar. Okay, and then a nitrogenous base here. Okay, so DNA is made up of 4 different types of nitrogenous bases. So the phosphate group and the sugar group, the carbon sugar group, is the same for every nucleotide, but then the base is what will differ. So there's 4 different types of bases, and the 4 different types are cytosine, and they all have a slightly different molecular structure. So there's cytosine, thymine, and then there's guanine, and then adenosine. Have you guys heard of these 4 different bases before in DNA?
No.
Okay, good. Okay, brand new information. So, so DNA, when it pairs together, you there's something, so DNA is double stranded, and sometimes you'll see DNA represented as dsDNA or double stranded DNA. So it is, the molecular structure of DNA is a helical structure. It's this double helix, we call it, which is, this is kind of my poor drawing of it. So, and then the backbone of the DNA here, which are these little blue lines, are the sugar, and then the little phosphate groups come off of that. And then within the center of it looks like a spiral ladder, kind of. And then within the spiral ladder, the bases, the nitrogenous bases, pair together. So, and they always pair, the cytosines and guanines always pair together, and then thymines and adenosine always pair together. One second. Okay, so I wanted to double check that because I didn't want to tell you guys, I was thinking of something else and I didn't want to give you guys bad information. Okay, so let me draw in, so I'll draw in the little bases here. And so just one for each color. So you'll have, along the way, you'll have cytosines in here, kind of randomly distributed, and then we'll have thymines. And each DNA strand has a particular sequence with it. And then we'll have guanines. Oops, let's do a different color for that. And then adenosines also are sprinkled in there. So along one strand, you'll have a certain sequence of bases, and then on the other strand, you'll have the pair for those sequences. So for cytosine, we'll have that pairing with guanine. And kind of so on and so forth. So that's DNA, okay? DNA will undergo a process called replication. And that's where the DNA just makes a copy of itself, okay? So that's called replication. When DNA gets transcribed into RNA, which is very similar to DNA, except instead of being a deoxyribonucleic acid, it is just a ribonucleic acid, and it's just a slightly different structure of the sugar group that's attached to the base. And then the other difference between RNA and DNA is that instead of thymine as one of the bases, it's another group called uracil. So RNA has something called uracil instead of thymine. Okay, so in a process called transcription, so instead of DNA replicating to make a copy of itself, sometimes DNA will be transcribed, or in a process called transcription. Into just a single-stranded, instead of a double-stranded, RNA molecule. And so it still has the bases attached to the sugar backbone, but it's just a single strand now. And instead of thymine, there's uracil. Okay, so we'll just kind of draw the different bases here. Okay, so, and this all happens, so the DNA lives inside the nucleus, and then it gets transcribed when we're ready to activate the blueprint. It gets transcribed into RNA. And actually, it's specifically called messenger RNA or mRNA. And it's like a little message from the nucleus that leaves the nucleus and goes out to the rest of the cell. So this all happens within the nucleus here. The mRNA then travels out to the cytoplasm. It leaves the nucleus and goes into the cytoplasm. And then it is translated. So instead of being transcribed, the term is now called translation. Okay, into what we call a protein. And proteins have their own structure. Instead of having being made up of these nucleotides like we talked about over here, the structure of proteins is made up of something called amino acids. So that's what this is over here. So it's a slightly different molecular structure, and they are also, instead of being double-stranded or single-stranded, they get folded and form these different sort of helical structures, or something called alpha sheets and beta sheets. And they kind of just look like a big blob once it's all folded up. It's still a single strand of amino acids all linked together in a chain, but then they fold together in a different structure than say the helical form of double-stranded DNA. All right, and so this is what we call dogma, meaning the central dogma. So the central dogma is kind of like the overarching formula for how life basically replicates itself with DNA, and then how it carries out its cellular processes through a process of transcription and translation. And this is preserved across all living species, from the plant or tree in your backyard to how your dog makes its fur, to how humans are able to carry out all the different metabolic processes that help keep us alive. So this is just kind of like the very most basic process. There's some other newer, relatively newer one. This was first described by two scientists, Watson and Crick, who won the Nobel Prize for discovering the helical structure of DNA, which was based on another scientist, Rosalind Franklin's, based on her research. And then since then, we have discovered, humans have discovered kind of alternate processes that sort of violate what we initially thought was a one-way process, where we always go have DNA that replicates itself, and then it gets transcribed to mRNA and then translated to protein. Sometimes you can have a process called reverse transcription, where you go from an RNA, a single-stranded RNA, to DNA. So that's called reverse transcription. And when you go from RNA to DNA, you create something called complementary DNA, or cDNA. And then sometimes you can have what are called RNA viruses that can replicate themselves, just go from RNA to RNA. You don't necessarily need to go from DNA to RNA. And some examples of RNA viruses would be the coronaviruses, like you guys, that's why we are doing the pop-up school in the first place, because there's a coronavirus and that's actually an RNA virus. So what the coronavirus does or what the coronavirus is made up of is RNA, okay? Let's see, and then some other examples of coronaviruses are like things like SARS or MERS, which I think were probably before you guys were even born. Those are examples of RNA viruses. And then we can also have something, in general, we think of the powerhouses of the cell, all of the action that gets, if you can think of DNA and RNA as blueprints, they're like the instruction manual, and then proteins as the workhorses of the cell. And in general, we think of proteins as being the molecular entities that carry out those cellular processes. But you can actually have something called non-coding RNA, or ncRNA. Like transfer RNAs or ribosomal RNAs, and they actually are more similar to proteins, even though they're made up of ribonucleic acids, because they actually carry out this process of translation, these little molecular machines. Let's see. Do you guys have questions about this stuff so far? Or anything you want me to go back and review that you're confused about?
No.
No, you got it? All right. So, and then the other thing I wanted to point out just about this overview about the central dogma of molecular biology is, I want to create some more room here. Okay. So there's another area of molecular biology that's relatively newer that is called epigenetics, which is a really exciting new field within genetics. And basically what it used to be that we thought whatever your DNA sequence was that you say that you're born with, that is what dictated what you looked like, and the expression of that DNA was just based strictly on whatever the blueprint was in your DNA. But there is another process called epigenetics. So epi meaning kind of on top of whatever this blueprint is. So if you have a DNA code, it's like, I don't know if any of you guys are into coding. So if you have a sequence of DNA of just different bases, okay. And so this is your DNA sequence, and then it gets transcribed into the corresponding RNA sequence here. There is a process of epigenetics that actually dictates which part of the code gets transcribed into RNA, okay? And the different ways that DNA is coiled up in the nucleus, and through a process called DNA methylation or histone modification, the DNA can either get more coiled and more bunched up and therefore harder to read and harder to express, or little molecular machines can actually carry out different chemical processes that allow the DNA to unwind and then make it easier to be read and transcribed and translated and then fully expressed. So this is how you have the exact same DNA in every cell in your body, but you have different cells being expressed in different ways. So let's take a cell in your hair. Okay, it's made or let's say a cell in your eye. A cell in your eye is going to look and act and behave and carry out different processes. That's very different than a cell, say, in your skin or in your muscle, for example. And so the blueprint is all the same. The code, the instruction manual is the same for every one of those cells. But then as you were developing, different epigenetic processes allowed certain parts of the DNA blueprint to be expressed and other parts to stay hidden. And that helps to explain how we have the same blueprint in every cell, but then drastically different expressions of it. All right, so that's probably plenty of information, especially coming at you for the first time. But this is kind of the very basic foundation for how living things replicate themselves and then also express themselves. All right. And so have you guys studied at all, are you learning about the animal kingdom and how there's different branches like bacteria and mammals and plants, and how they make up different branches of the phylogenetic tree? Or learned anything about DNA before? This is like very first time.
Very first time.
Very first time.
All right. I'm just curious, what are your guys' favorite subjects in school so far?
Drama.
Oh, okay, cool.
What kind of school are you talking about?
It could be, well, any subject.
Mathematics.
Okay.
Same here.
Oh, nice. And what are you guys learning in mathematics right now?
Here. So we're both in the same class. So, we were just.
I said math, not math.
Oh, I thought it was math. You said that.
I'm learning math and mathematics.
All right.
Well, math for me, and I forget what we're learning in math at school right now. It's been so long.
There's a lot going on right now. Yeah.
It's
So
been 3 weeks.
yeah. So one way to think about biochemistry and how it relates to mathematics is, and really all the sciences, is that biochemistry, which is a combination of biology and chemistry, they all kind of build up on top of each other. So what I would say is most foundational is mathematics. So kind of the underlying order to everything, all of the sciences, is math. And then on top of that, on top of math, you have physics. And so in high school, you'll learn a little bit more about physics and all the different laws of physics, like thermodynamics or gravity and all that kind of stuff. And on top of physics, you get chemistry, okay, which are all the different ways that chemicals interact with each other and react. And then biology is on top of that. So they're all related to each other. In school, the subjects get artificially or arbitrarily separated. We separate them into different subjects to learn, but it's important, actually more interesting, to really think of them all being connected to each other. And so a lot of what we study in biochemistry as a chemical process, let's say the chemical process of DNA replicating itself, that actually is all a highly ordered process that still follows the same laws of physics and mathematics, but just at a higher order level, on the level of an organism.
Wait, so biochemistry is a mix between biography and chemistry?
Biology and chemistry.
Oh.
Yeah. So
I heard biography for some reason.
Yeah. So you can think of, so if biology is the study of all living organisms, okay, then, and chemistry you can have in non-living organisms or non-living entities, and then or living organisms, right? So you can have the like a chemical reaction that happens in let's say like a combustion engine or something like that. Or you can have chemical processes that are happening in a living organism like the human body. And so what the field of biochemistry is looking at the chemical processes within biological organisms. And so it's really, and it's a little bit messier than some of the other sciences like physics or something like strict mathematics, because biological organisms are just infinitely complex. There's so many processes going on that are hard to account for and that we're constantly discovering, which makes it both challenging but also really interesting. And you will probably, I would say, like anybody who works in the life sciences, so anything related to health or medicine or anything really related to, if you're working with living organisms, you'll probably learn more about, have some sort of biochemistry background in that. And then did you guys know any doctors or any surgeons or anything like that? Or you have any questions about that at all?
My grandma was a nurse.
Oh, okay, cool. Who's holding their hand up?
Me, my second cousin is a surgeon.
Oh, cool.
Nurse, I think. Yeah, surgeon. Yeah, my second cousin's a surgeon. My mom's cousin.
Oh, very cool. Very cool. All right. And then, all right. So if you guys don't have any other questions, that's it for my lesson for today. And yeah, if you guys wanted to learn more about something specifically, like some sort of biochemistry related to some, yeah, what's that?
What do you think might be the solution to coronavirus?
Oh my, okay, so that's a big question. So I think actually a lot of what the solutions will be definitely involve biochemistry and different scientists who work with biochemical processes. So let's say, I think part of the solution will be developing a vaccine for coronavirus. And so in order to, a lot of vaccines actually, some of the newer vaccines that are more rapidly, that we can develop more rapidly, are actually messenger RNA vaccines. And so what scientists will do is look at the RNA sequence of, let's say, coronavirus, and then you can generate a messenger RNA-based vaccine that you can then give to people, and then in a very small amount, and then your body can develop its own, can learn what that viral sequence looks like, and then develop machinery with your immune cells can develop machinery that match that sequence, and then can quickly attack and defeat the virus before it makes you really sick. So that's going to be definitely one part of the solution to coronavirus. Another major part that I think will help a lot is to have more widespread testing of people. So part of the reason why we're all cooped up and not allowed to go to school or go to work is because
they're not testing enough.
Yeah, so exactly. And so part of what makes testing difficult is one problem we're running into is how rapidly we can get a result. And so there are some testing protocols that take a couple of days to get results back. And so when you're talking about a problem on the scale of coronavirus, to have a 2 or 3 day delay on results is too long. It's allowing people to spread the virus unknowingly without knowing that they're positive. And then even the more rapid solutions, where you can get a result back in 15 minutes, some of the actual testing kits, they can only process one sample at a time. And so if you're only that's, one sample every 15 minutes, say, and that's only 4 samples per hour for that one machine, it's just not a high enough throughput. And so some of the faster testing methods incorporate something called PCR, polymerase chain reaction, which is based on this exact, is related directly to the lesson that I just taught you guys. So it's a process of reverse transcribing and then transcribing many different sequences of either DNA or RNA, and you can amplify a sequence enough to get a positive result on a very small sample. So that's another challenge with testing too, is getting, being able to get enough of a DNA sequence from something, or RNA sequence, from a small sample. So from a lot of people, you can take a sample of saliva or a small sample of blood and then isolate whatever marker you're trying to pick up that gives you either a positive or a negative for your test result. And so, yeah, through this process of, we've been able to basically mimic what we've learned happens at a cellular level. We can do that now in a lab with all the same little molecular machines. So yeah, that'll be part of the response to coronavirus too. One is developing a vaccine, and the other one is just more rapid turnaround for tests and more widespread testing. And I think a lot of the testing issues we've run into relate to supply chain stuff, getting the right, you need the right, every science, these are like a bunch of different little science experiments. Every time we run a test on someone, and every science experiment is like a cooking recipe. You need all your different little ingredients in order for the test to run properly. And so we're running into some issues with that too. But I think that we just need to give it a little bit more time, and hopefully in a couple weeks we'll have something better. That's a good question though. Anything else? Anything else you guys are curious about with coronavirus and how it relates to biochemistry? Do you guys know, so one kind of interesting fact about coronaviruses too is that they typically come from animals. So
apparently my dad heard somebody ate a bat.
Yeah, that's one theory for what happened. So this latest coronavirus is traced back to bats, and bats are actually thought to be the host. So they don't necessarily get sick, but they are able to host the virus and spread it. And so whenever we see epidemics like we see with coronavirus, and then we saw a couple years ago with MERS or SARS, it comes up every 10 to 15 years or so, is when the new virus kind of hops to a different species. And then we have no prior immunity, no exposure to this, and so it just spreads really quickly. But part of the reason why these viruses can jump from, say, from a different species like a bat all the way over to humans is because we all share the same DNA, which is kind of crazy when you think about it. So I think, we share over 90% of our DNA with other animals. We're all very similar in that way.
Good to know.
All right. I think I've maxed out your guys' attention span. It's been really fun. And then, yeah, I can also put together a lesson about, specifically about how viruses work too, if you're curious about viruses. Viruses are different from other living organisms because they don't have the ability to replicate themselves. So when we talk about DNA undergoing its own replication, all of the machinery for that process, we living organisms have that kind of machinery. That's what's part of what's encoded in our DNA. But viruses are made up of, don't have that machinery. They only have their genetic code, the blueprint or the instruction manual, and then they're much simpler. They're just encased in a protein envelope. So they need a host to use, they need to hack into the host and steal machinery so that they can replicate. And so viruses are kind of unique in that way. Not unlike a virus for a computer, let's say. All right.
Does anybody else have any questions for Tracy before she leaves us?
No.
Okay. Well, thank you so much for joining us today. This was very special, and yeah, I think we all learned something new today, so that's very neat.